Okay, oops, that's the end. Uh-oh, you guys just saw the whole thing. Okay, so today we're doing import to the mitochondria. We're gonna finish off the nucleus quickly. Just a reminder, if you were going to write an exam question, you have till today 5 p.m. to get it to me. I've gotten a few good ones. I've gotten some that I think people forgot that it had to be multiple choice. So that's a big one. It has to be multiple choice, and it has to be good. If it's good and it can be fixed, I'll still give you a point. But if it's perfect, you can get 4 points. So that's a big deal. Also, I meant to say this the other day. I forgot. Sorry. If you're a Friday recitation, you want to come today because you think you're going to get something out of today's recitation that might help you for your exam. Feel free to come. And I think that's it for business. So far we've talked about how we get many proteins from one gene through processing of RNA, protein transport. Specifically, we went through the secretory pathway. I got a few questions that made me a little bit worried. I want to make sure everyone understands that the secretory pathway is not only secretion, it's the pathway that the default is secretion, but it can go to the lysosome or the plasma membrane too. I want everyone to understand that. It's not just secreted proteins. And now, the other day we started the nucleus, about import and export from the nucleus with importance and RAN. And not to confuse RAN with RAB. Remember, we learned about RAB, totally different from RAN. Another easy point to be confused on. So today we're going to finish that up and then do the mitochondria. We're going to do the mitochondria. Anyways, so this is where we stopped. We were talking about import of proteins into the nucleus by important or an import receptor recognizing the NLS in a protein, nuclear localization signal in a protein, and how RAN GTP allows for release of that cargo once it gets to the nucleus. And this is how it works. Here's your important. I'm going to turn it off because there's some red that I want you to see. Okay, so here's your important, this S-shaped thing, and it's also here if you don't like the protein shape. And so you have two binding sites, one binding site for the cargo protein and one for RAN GTP. And basically what this is showing you is that when your cargo protein's there, there's this red loop here that interacts with the NLS in your cargo protein. But when RAN binds, this loop it flips. So there's a conformational change that happens upon binding of RAN. And so now the part of the protein that interacts with the NLS is taken away because it's interacting with RAN. So what's going to happen if you take away that part of the protein? You're not going to bind to your cargo anymore. And that's how the cargo is dropped off in the nucleus. And so I really like this. It's a very nice, simple mechanism to understand how RAN participates in the release of the cargo in the nucleus. Okay, any questions about that concept? Yes?
Would you say the red protein was in the first compound?
Here?
To the left.
Here?
Yeah.
So there's RAN, and then there's your binding site for your cargo protein, and there's this red loop, which is part of your important. This is your important. This whole S thing is your important, and it has a loop. What I mean by a loop, I mean the amino acids and the way they fold form a loop that's flexible, and it moves. And when the cargo is there, that loop's going to interact. Those amino acids in that loop are going to interact with the NLS. But when RAN binds, there's a conformational change and it flips the loop around. And so now it's not able to bind to the cargo anymore, and that's what causes it to release it. Okay? Any other questions about this? Okay, very good. Okay. So now that we understand nuclear localization, we can think about regulation of nuclear localization, right? What's the key to getting a protein into the nucleus? Recognition, a recognition of what? NLS, right? Okay. So if you hide your NLS, will you go into the nucleus? No, because you can't interact with your important. And so in signaling, this happens. So just to take one step back, what kind of proteins go into the nucleus? Proteins involved with transcription. Specifically, what kind of proteins? So there can be proteins involved with the machinery involved with transcription, like RNA polymerase, but there's also proteins involved with whether or not a gene is going to be transcribed. What are those proteins called? Does anyone know what I'm getting at? Oh, well. Are you observing? Dr. Wimmers likes my lecture style. It's right there. No, stay. I'm going to do a dance soon. Right, I do a dance every day, don't I? So what kind of proteins affect whether or not a gene is going to be transcribed? Yes. Yeah, exactly. Transcription factors, right? So whether or not a gene is going to be transcribed is frequently regulated by whether or not that transcription factor makes it into the nucleus. If it's not in the nucleus, it can't bind to DNA. If it can't bind to DNA, it can't help recruit RNA polymerase. If it can't recruit RNA polymerase, you can't get transcription, can't get RNA, can't get protein, can't get gene expression. Does everyone follow that? Okay, so so this is one way. There are many ways that transcription factors are regulated, and when we go into signaling next week, we're going to start to learn about that, because signaling is all about something binding to a receptor, a whole cascade of phosphorylation that sends a transcription factor into the nucleus in some way or other to activate gene expression, right? Remember, cells respond to their environment, and their environment is sensed by things binding to receptors. Does that make sense? A little bit? Okay. So this example is showing this one transcription factor, and it is phosphorylated. And when it's phosphorylated, its nuclear import signal is hidden, right? You have these lysines and arginines that need to be bound by your important. Well, if you phosphorylate them, then what's going to happen? Is it going to be able to bind to your important? No, right? So it's excluded from the nucleus. And in certain situations, in this case, in activated T cells, you'll have signaling that will result in a release in calcium, and that's involved in signaling. And it will actually activate binding of a phosphatase, which will remove those phosphates and expose the transcription factor. Okay? Expose the NLS. Right? So now the NLS is able to bind to the important, which it's not even showing in this picture. And so the important brings it into the nucleus. It does what it needs to do, which is usually bind DNA and activate gene expression. And then when it needs to be turned off, calcium will go down. And then that will inactivate this phosphatase, which will allow a kinase to be activated and rephosphorylate that NLS. And then a nuclear export signal will become exposed. And so I guess I forgot to tell you that the phosphatase binds and covers up the nuclear export signal. So the nuclear export signal will be exposed, and then it will be exported out of the nucleus. And that's a very nice way to regulate gene expression. Allow your transcription factor into the nucleus by allowing access to the NLS, and then under other conditions, phosphorylate that NLS and allow exposure of the nuclear export signal, and then it can go out. Oh dear. There's so many dead eyes. It's like zombies in here. I'm gonna guess it's because everyone's been studying really hard and you're all tired. Yes?
So where is the transcription factor? Is it on the NLS or is it somewhere else?
Okay, thank you. I love these questions. Where is the transcription factor? This protein is the transcription factor, and fat is the transcription factor. It is a protein that has a sequence in it that is an NLS. Okay? So an NLS is not a protein. An NLS is a sequence within a protein. It's a kind of like a signal sequence. It's a type of signal sequence. It's not cleaved. It's a type of, you can think of it as a type of signal sequence that's going to direct a protein to a certain place. Thank you for that question. Any other questions? Yes.
Why was it phosphorylated in the first place? Like, 'cause I know you have to take off the phosphates, but why was it phosphorylated?
It stays phosphorylated to keep it out of the nucleus. So it's always phosphorylated except for when you activate a phosphatase. So the question is, why is it phosphorylated? That's how it is. That's how it's regulated. It is phosphorylated, and when it's dephosphorylated by activating a phosphatase, then it exposes the NLS and it goes in. Yes.
So the difference in calcium concentrations there doesn't have anything to do with the fact that in one part it's in the cytosol and the other part it's in the nucleus.
That's what regulates it.
Okay, so but it's saying activated versus resting.
Yeah, so in an activated T cell, so I didn't want to go too much into the immunology because that can get confusing for people who haven't taken it or don't care about it. But so in your T cell, you can have a resting T cell that where it hasn't been exposed to any antigen in any way. And then you can get an activated T cell where it's met up with MHC containing peptide, displaying peptide that says there's an infected something here. And what happens to a T cell when it gets activated? What's one example of something that needs to happen when you have an activated T cell? Anyone who's taken immunology or likes immunology or can just think about what activation might do to a T cell. What do T cells do? Huh? They send signals. Okay, that's one thing they do. So they secrete cytokines to alert other cells of things. What else do they do? So T cells are T cells, B cells are B cells, but definitely T cells and B cells come from a similar lineage. But T cells, there are different kinds of T cells, but T cells themselves, what they do is if they're CD8 T cells, they're cytotoxic T cells, so they kill virally infected cells. If they're CD4 T cells, then they are helper T cells. If you guys have ever learned about HIV, you may have heard about these things. Helper T cells will activate B cells, which will then secrete antibody. They do a whole lot of things, but one main thing that needs to happen when they're activated is they need to explode in numbers. They need to proliferate like crazy. So one activated T cell, it makes sense, right? If you're a T cell and you've seen something that is pissing you off, you need to get your peeps, go attack, right? You need to explode in your numbers so you can go and kill virally infected cells, kill cancer cells, or activate lots of B cells. So let's go back to activation of T cells, right? So to activate, and then you need to replicate. So now you have to talk about going into S phase, right? Synthesizing DNA, copying your DNA. So there's a lot of gene expression with moving forward in your cell cycle. Mitosis, right? If you want to proliferate, which means expand your numbers, you need to go through mitosis. So lots of gene expression, right? So that's what we're talking about here when we're talking about an activated T cell. So when you have an activated T cell, there's signaling from receptors, which results in increased calcium, which then results in activation of this phosphatase, which then removes these phosphates, which then exposes your nuclear import signal, goes in, activates all those things that need to happen so that you can be an activated T cell, multiply, copy your DNA, go through mitosis, and just explode in numbers, and then make cytokines, and I don't know, lots of things that T cells need to do. But that can't stay on forever. What happens if you have an uncontrolled number of T cells? You could have some autoimmune issues. And also the other thing is whenever you have unregulated growth of cells, you should start thinking cancer, right? I guess often B cell. There's T cell lymphomas, yeah. Leukemia, and so you need to shut it down, right? So you need to turn off all that signal. All that signaling that caused an expansion of your T cell numbers needs to be shut off, and you need to not only. Usually, what you end up doing is killing yourself. Well, not yourself, but your T cells. But the other thing you need to do is not keep making more, right? So you shut off what you have, and that's where your calcium levels are going to drop, and you're going to have your calcium levels are going to drop, and you're going to activate your kinase, and you're going to phosphorylate your NLS, and you're going to expose your nuclear export signal. You're going to be kicked out of your nucleus, and you're not going to be able to get back in. Did everyone follow that story? No. Who said no?
I said no.
Thank you. I love
Like,
honesty.
you don't really have to explain it again because the point was explaining. It was just a lot of other stuff that.
Yeah, I was trying to give you context. Yeah. Did the context confuse people? A little bit. Okay.
So it gets rephosphorylated in the nucleus, and then that's what makes it.
Well,
Or that exposes the exports.
No, there's two things. So one is the NLS, which is here, and one is the export signal, which is here. So the export signal is covered up here because your phosphatase binds there, and it comes in, and so that's covered up the whole time. But the decrease in the calcium is going to kick that off, and then that's going to also allow. So now your phosphatase is gone, which is going to allow you to be phosphorylated again, but also allow exposure of your nuclear export signal. Make sense?
So it's calcium regulating.
Calcium regulated. Yep. Questions? Comments? Feelings? Emotions? Feelings and emotions are the same thing. Right. How's everyone doing? Tired. I know, it's the weather. I blame the weather and the lack of sunlight as the day goes on. Okay, let's move on. Practice problem. You guys are going to chit-chat with your friends for a little bit. You are studying a viral protein that appears to be facilitating the import of certain proteins into the nucleus. Design an experiment to test your hypothesis. So basically, you think this protein is facilitating nuclear import. So what are you going to do to see whether or not your protein is facilitating nuclear import? Another viral protein appears to be preventing export. What could be going on here? How would you test your hypothesis? So stop and chat for a few minutes until you start talking about your weekend plans.
That doesn't really tell you when
Am I seeing like a tie-in with the I thought that was what was kind of it was like export the vote.
I think they do it too, man.
Like I understand this and I understand brand GTP design brand
This wasn't an example at all because I wasn't showing you anything about importance or moving of anything. I was just showing you the acts of interaction. That's all that I was showing you. So not you didn't see anything about what would bring that's all there. Right? So if you have your analysis closed, what's going to bind important. And it's going to go in that, and then brands and GTP is going to bind. same thing is all there. That's just an example. I gave you like bare bones explanation of import with like the reality involved, and then I gave you a real description factors on how that is regulated by the strong exposure of
So brand, the day was more of a mechanism, and this is an example.
from other sports.
Right.
And this is just an example of import and how regulation of whether or not you
had organic
your
A lot of people want to figure out how to just one midterm and one final.
what might that
Yeah, so with like 6 cells and part of those with calcium or bonds. So we still that would stop it altogether. Stop.
the other is where and whenever you share all
Right.
She took it this morning. Yeah, I took it this morning. I was pretty good.
no one thinks about what so I guess
So preventing nuclear export is not letting the calcium levels go down. It's not just letting the kinase come in.
Because whatever I did did not work out. Like, he said he took them from the book, but like the ones in our book, that's what I didn't know I had to
that one
But something regulates dephosphorylation
whatever I did, something. We now see them. We have like we have like a
and so
Which is a component of when it's inside the needs to drop down. And then that activates and I guess like when you phosphorylate it, I think it's a lot
is that okay, guys? Do you guys need another minute? Are you guys still fleshing out ideas, or do you want to move forward with explanations? We can move forward. I got approval. Okay. Okay, so I got some good questions while I got two, actually I got a good clarification question. Two good clarification questions while this was going on. People who were kind of stuck on the last thing. So I wanted to clarify two things. Someone asked me, wait a minute. Wait, is this calcium regulated or is this NLS regulated? I don't understand where it was all coming together. And that was one, and then I got other examples, kind of other questions about that. And so what that made me realize is you need to understand so many different steps in the pathway. So, yes, so that slide I just showed you is one example of one transcription factor. Not all transcription factors are regulated by phosphorylation. Not all have calcium involved in any way. This is one example that all talks about access to nuclear localization signals. One example. And yes, it's calcium regulated in this example because calcium regulates the activity of calcineurin, which is the phosphatase. Okay? And yes, it's NLS regulated because it all has to do with whether or not the NLS is exposed. The other related question, someone said, well, what about RAN? Where does that all fit in? And that was a great one. So here I didn't talk about RAN at all, right? But it's here because here I'm only talking about the NLS and whether or not you can see the NLS. But you need to think to yourself, well, okay, well, what does the NLS bind to? it binds to an important, and then it goes in through a nuclear pore. But then it needs to be released inside the nucleus. What does that? RAN-GTP, right. So just start to try to piece this all together, and it'll start to make a lot more sense. Okay, so let's go to this. So then I got a question about this that was like, what are you talking about? And I appreciate those kind of questions. So what I'm asking about is, if you have a protein and it affects whether or not another protein goes into the nucleus, what could it be doing to force a protein into the nucleus? And so then we started talking about phosphorylation and stuff, and I was like, well, don't forget the example I just gave you was one transcription factor. So some transcription factors are never phosphorylated. A lot are, but some never are. So I just want you to keep that. So does anyone want to give me an idea of what they were thinking about? Start a little discussion. Anyone? Yes.
So for the first question, I was thinking that maybe the viral protein is binding to those proteins in such a way that they actually show the nuclear localization space and can bind to it.
Okay. So I heard two things. One I heard is maybe the viral protein is binding to another protein in a way that it exposes a nuclear localization signal and allows interaction with an important. So that is one thing. Let's say a viral protein binds to a protein and causes a conformational change exposing an NLS, or even more so, let's say a viral protein binds to a protein that doesn't even have an NLS, but the viral protein has an NLS. That could force interaction with an important. And so some protein can piggyback its way in. The other thing that can happen is sometimes viruses use that way to get into a nucleus. Let's say a viral protein has no NLS. It'll piggyback on a cellular protein with an NLS. So those were some very good ideas. Anyone want to add to that? So what would you do to test that? If you were working in the lab and you say, my viral protein must be somehow exposing an NLS on this transcription factor, what's the first step that you would do? Well, probably the first thing you need to do is see if there is an NLS. Yeah.
I was gonna say you could like tag the proteins and then see where they were fast.
Okay, so you can tag the proteins, make them fluorescent colors, and see with and without your protein, with the viral protein, where is the transcription factor. Put in transcription factor, does it go to the nucleus? That's a good way to see if your viral protein is forcing something into the nucleus. But how would you test the mechanism? I guess that's a lot harder. Maybe we won't go there. Because that might... Yes?
Is this are these certain proteins, are they not supposed to be going into the nucleus?
You can design any scenario you want, really.
Because if they were proteins that were going to be going into the nucleus anyway, how would we detect any
Right, so you wouldn't have the question if it was going to go into the nucleus anyways. The question wouldn't be there because you would never see that the virus was doing anything. But let's say it was a protein that normally doesn't go into the nucleus and it suddenly is in. Or let's say it's a protein that normally the NLS is phosphorylated, like the example we just had, and suddenly it's in. Right? So you can put the viral protein in and check to see if your transcription factor is not phosphorylated anymore. Right? So I'm just trying to get you guys to think about how to use this information that we're going through. Does anyone want to add to that? Did I clarify?
Yeah.
Okay. Yes.
Can you explain the possible like piggybacking? Say the protein does not have an NLS, but it binds to a protein with an NLS.
Right. So if you don't have an NLS and a viral protein has an NLS, it can force a protein into the nucleus. How would that work? Yes.
And just find a way to bind to the protein, and then it would bind the important.
Yeah, exactly. So it would just basically be that a protein would have some kind of affinity for whatever protein it needs to get into the nucleus. and it would have its own NLS and interact with an import and go in the same exact way. And the way you can test that is you can look to see, do these proteins interact? What happens if I mutate something, they don't interact? Is it not going into the nucleus anymore? Stuff like that. Any other questions? Okay. So here's another one. Another viral protein appears to be preventing nuclear export of a protein. What could be going on here? Preventing nuclear export. What might be happening? Yep.
Could it possibly be like a competitive inhibitor kind of situation that it would prevent the cargo from ever going on the export in the first place?
So it's preventing the cargo from being exported. How? Yes.
The most two likely ways would be binding to exportin itself, so it can't bind to a protein, or messing with the guanine exchange factor.
Okay, so binding to an exportin, so it can't bind to the protein that needs to get out. Messing with the GEF, so that, well, that's really bad, isn't it? Messing with the GEF so that you can't activate RAN at all. Yep.
Binding to the nuclear export signal on the cargo itself so that the cargo can't bind to it.
Binding to the nuclear export
So that
signal.
The cargo can't bind to the import.
Right, binding to nuclear export signal so the cargo can't bind to the exportin. Right, so that would be the most specific way, because then you can target one protein. If you mess with a GEF, you mess with all import and export, and that's a good way to kill someone. So that wouldn't be in the best interest of a virus. Okay, one more. I saw a hand somewhere else. No? Okay, so we'll move forward. I just wanted to get you guys thinking about using this information because that's how I like to design questions. Okay, so let's spend a few minutes on import into the mitochondria. So obviously, it's more of the same story, right? Different proteins reside in different places because they need to do certain things and they need to get to certain places to do their job. Key players, again, signal sequence, translocators, chaperones, and energy. Does this sound familiar? It seems to be a theme we keep hearing. Okay, so for mitochondria, they're post-translationally directed. So you're never going to have co-translational import into the mitochondria. Always post-translationally. And it recognizes a signal, but it's not a specific amino acid sequence. It's a structure. It's a sequence with the ability to fold into a helix, which has positively charged amino acids on one side and hydrophobic amino acids on the other. Okay, so it's a structure that's recognized by a receptor, and the signal sequence is cleaved by signal peptidase also. And then you have translocation through translocons. So this should be very reminiscent of the ER2 guys. And the only difference is you have two membranes to deal with. So you have Tim and Tom. I love it, right? Inner membrane, outer membrane. So you should never get confused as to which one is where, right? So, and it stands for translocase of the outer membrane and translocase of the inner membrane. And then there's also some other ones that aren't as easy to remember, but basically Tom is the one on the outside. So major steps of import. Translation in the cytosol because it's post-translationally imported. And then it's bound by chaperones, just kind of like when you think about post-translational import into the ER, bound by chaperones to keep from folding and aggregating. And then the signal sequence is recognized by a receptor that's associated with the Tom complex. So that's shown right here. So you have recognition of the signal sequence, which is right next to the translocon, and then it gets inserted into the membrane, and it actually goes all the way through Tom and Tim into the matrix. And then on the inside, there's a signal peptidase that will cleave off the signal sequence, and there you have it. That's a protein that's going into the matrix. Yes?
And like 2 slides before, I thought you said that it's a structure that's recognized and not.
Yes, the signal sequence is actually a structure rather than like lysine, arginine. So by structure, I mean amino acids in a protein fold into certain structures. And so it will fold into a helix that has, what did I say? Positively charged on one side and hydrophobic on the other side. And that's what's going to allow interaction with that receptor. Does that make sense? You guess? Come see me. Questions about this? It's very similar to ER up to this point. Yes?
And this is because most of the mitochondrial proteins are being encoded in the nucleus rather than in the mitochondrial genome.
Yes. 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 Well, let's see. Do I? Yes. I'm not really talking about anything else. Yes. Let's go with that. Yes. You're making me want to. I'm not going to expand. Okay. So that's what we just talked about. Okay, so ATP hydrolysis in the form of HSP70 bound ATP and a pH gradient, which is on the membrane of the inner membrane of the mitochondria. That's what drives transport across the mitochondrial membrane. So you have your HSP70 bound to your protein that binds to the receptor, and then it's going to be fed through two translocons, and that takes some energy. And in terms of binding and release by HSP70, and there are two HSP70s to keep track of. There's some on the outside, azolic HSP70, and there's mitochondrial HSP70. So HSP70 that lives in the mitochondria. So they're both involved with pushing and pulling kind of into the matrix. And then there's this pH gradient, not pH, this pH gradient, which is involved in, where's that come from? You guys know about that electrochemical gradient on the across the inner membrane? Do you know where that comes from? What does the mitochondria do?
Makes ATP.
ATP. So remember electron transport and all that stuff that happens? That's not separate. That's not a different mitochondria. It's the same mitochondria that proteins need to come into. I correct myself up. So that is also involved with pulling the protein in. Okay. So I'm talking about HSP70. How does HSP70 drive import, right? So you have the cytosolic HSP70 that's binding to the protein and preventing folding and aggregation, right? But you also have the mitochondrial HSP70 that's helping to bring the protein in, and that works in. There are two theories on how HSP70 pulls the protein into the mitochondria. And so one is called the thermal ratchet mechanism. And that is where you've got this thermal motion. You know how there's thermal motion, constant motion happening. So you can imagine your protein in your translocon, and what happens is mitochondrial HSP70 on the inside, it will bind it, and as that protein is moving, another one will latch on. So it's like a hand over hand pulling in, and it's just using that thermal energy and then kind of prevent the binding of HSP70s to proteins, preventing it from slipping back up. Okay? And then it's released through ATP hydrolysis here. And so then there's the cross-bridge ratchet model, where it's actually hypothesized that HSP70 conformational change that happens just because when you have ATP hydrolysis with HSP70, it changes the conformation of HSP70, and it's actually pulling down, actively pulling down on the protein to bring it in. And so here you can see it's bound, you have ATP hydrolysis, and it pulls down. And then it comes off, and another one's going to bind, ATP hydrolysis, pull down, come up. So one's a pulling down motion, and another is like as the protein kind of comes in, another one's going to bind and preventing it from slipping back up until it's all the way in. One's a little bit more passive. questions about this? Should we just call it quits? No, I think we have another one. Okay. So I told you import into the inner membrane or intermembrane space. So I told you about getting into the matrix, but also what if a protein needs to be a membrane protein in the inner membrane, right? Or what if it needs to live in the intermembrane space? So there are different ways that happens. And usually what happens is you'll have import all the way through, so in through Tom and Tim into the matrix, and then you'll get cleavage. So this is a membrane protein. You'll get cleavage of your signal sequence, but you'll also have a stop transfer sequence, and that'll get moved over into the membrane, and then it'll finish getting pulled in through Tom. So that's how it ends up being an intermembrane protein. And you can also have another situation where you'll have import all the way into the matrix, and you'll have cleavage of your signal sequence, and you'll be in the matrix, and then there'll be another signal sequence which will target it to another translocon called the OXA complex, and then it'll be recognized and moved over into the membrane. So one is where it never makes it all the way into the matrix and just gets moved over, and the other one is where it makes it all the way in and another signal sequence targets it to a translocon that puts it up on the inner membrane. And then the other one is similar, where you'll have it in your inner membrane, and then a protease is in there and it cleaves it off, and that's how you have a protein that lives in that intermembrane space. And then there's the other multi-pass transmembrane proteins where you can have chaperones bind. I'm not going to ask you for every single mechanism, so do not freak out. I just want you to understand, just like we went through those integral membrane proteins, there's many types of proteins. Some are in the membrane, some have N-terminal, C-terminal, some are multi-pass. Remember we went through all that? Well, it's similar here. You also have two membranes to deal with. So you have to think about how when you have import of a protein into an organelle with two membranes, some proteins are going to function in the matrix, some are going to function in the intermembrane space, some are going to be membrane proteins. Just appreciate that and understand that there are two translocons, and there are these different ways that can get proteins into different places. And then, of course, there's these porins. Porins are pores that are in the mitochondrial outer membrane that allow ions and small molecules to pass through. And those are actually translated and then go through the Tom complex, so they're in the intermembrane space and bound by chaperones. And then there's a SAM complex that actually assembles them because they're beta barrel proteins. And they can't get into a membrane in the same way. They don't have that like one little helical transmembrane domain. They actually are this huge beta barrel, and they're assembled by this other complex in the membrane. And so something that might be interesting to you guys or not, if you're interested in the embosymbiotic theory, is that this complex, the SAM complex, is conserved in eukaryotic cells and bacteria, so that's more evidence supporting the whole embosymbiotic origin of mitochondria. Is it even a theory, or it's just believed at this point? Okay, I think that's it. Yep. Any questions? We have 5 minutes. Don't go away if you don't want to.